Let's begin our look at the physiology of muscle contraction. And we'll start with the events at the neuromuscular junction or the motor end plate. There are two major events that take place, and we'll look at the details of these events in just a second. The first is that an action potential is created in the somatic motor neuron, and then that action potential causes the release of acetylcholine from the axon terminal, which then stimulates skeletal muscle contraction. So really here we're looking at the events that are going on in the neuron. So the events at the neuromuscular junction then set off which are here, these are the events at the neuromuscular junction in this figure, or in this flowchart here, then set off this chain of events within the skeletal muscle cell itself that are going to lead to that muscle cell contracting. So let's take a look at what's going on at the neuromuscular junction. So the first event that happens here is that an action potential that's spreading down the axon arrives at the axon terminal. Now remember that the action potential, being a voltage change, is going to stimulate or cause these voltage-gated calcium channels that are located in the membrane of the axon terminal to open. And when these voltage-gated calcium channels open, calcium is going to enter the axon terminal. So this term voltage-gated here implies that these calcium channels are sensitive to changes in membrane voltage, so changes in membrane voltage like action potentials. So here calcium is causing the synaptic vesicles to release their neurotransmitter by exocytosis into the synaptic cleft. So recall that the synaptic vesicles, which contain acetylcholine, are just essentially kind of stationed within the axon terminal waiting for the stimulus to release them. And then that stimulus is calcium entering the axon terminal, and calcium actually assists in the synaptic vesicles binding with the plasma membrane of the axon terminal, fusing with it, and essentially dumping their acetylcholine cargo into the synaptic cleft, which is the space between the axon terminal and the sarcolemma of the skeletal muscle. So those are the first three events here that happen at the neuromuscular junction. Picking up from that third event where calcium entry causes acetylcholine to be released by exocytosis into the synaptic cleft or the neuromuscular junction there. So once released, from the axon terminal, the acetylcholine will diffuse across the synaptic cleft, and it will actually bind to those ACH receptors that I pointed out in a previous lecture segment. These ACH receptors are on the sarcolemma surface, and so actually they're embedded within the plasma membrane in the junctional folds of the neuromuscular junction here. And ACH receptors are only found at the opposite end of an axon terminal here, because these are the only places where acetylcholine is released. And so they only show a handful of them here. There's only about 5 or so acetylcholine receptors here, but usually there's thousands of these in a single neuromuscular junction. So this diagram is a little bit simplified, but it gives you the general idea that there are acetylcholine receptors there. So it turns out that the acetylcholine receptor is actually an ion channel, which means that when acetylcholine is not present, it is closed. But when acetylcholine is present and is bound to the channel, that channel then opens, and it allows sodium to diffuse into our muscle cell and potassium to diffuse out of the muscle cell. And typically what we see is that we see more sodium diffusing in than we see potassium diffusing out. And so this actually causes a voltage change inside of our sarcolemma, and that is inside of our plasma membrane at our neuromuscular junction. And this voltage change is referred to as an end plate potential. So here's another phrase that uses that word potential here. So what I want you to think here is when you hear the word potential, think voltage. It's a voltage change. So we saw an action potential that travels down the axon to the axon terminal. That action potential is really just a voltage change in the plasma membrane. This end plate potential is also a voltage change. So I want you to think anytime that you hear the word potential, at least in the context of neuromuscular physiology, I want you to think about voltage changes. So that's what the term potential is referring to here. Okay, so we see voltage change occurring because this acetylcholine receptor has opened and allowed sodium to diffuse into the cell and potassium to diffuse out. So since sodium diffused in, the voltage change that occurs here is a more positive voltage change. So that is because sodium is a positively charged ion. When it enters the cell here, it makes the inside of the sarcolemma at this point in the cell a little bit more positive. So we refer to this as a depolarization because the voltage becomes less negative, right, because a positively charged substance is entering our cell here. So the voltage inside of the membrane here would be depolarizing. That is, it would become more positive for a brief time. So after a few moments, really a few thousandths of a second is really all it takes, is that the acetylcholine that was released by our axon terminal here is very quickly broken down by an enzyme that's called acetylcholinesterase. And acetylcholinesterase effectively inactivates acetylcholine, so that way signaling stops almost as soon as it begins, and this really helps prevent the muscle from becoming overstimulated. So after this end plate potential occurs here, or this local voltage change in our skeletal muscle, what we see next is a series of events that are called excitation occurring in the skeletal muscle, and excitation will ultimately lead to muscle contraction.